Hello and welcome everybody to my channel ominibioentrance, and today we will be seeing CSIR PYQs of June 2023 Shift 1. I will be dealing with applied biology and methodology, DNA replication, protein synthesis and genetics. Coming to part C, the first question. Following statements are about the features of immunoassays used to assay biomolecules. Radioimmunoassays are more sensitive than enzyme-linked immunosorbent assays with chromogenic substrate. Now they have asked for combination of all correct statements. So A is correct. ELISAs with chromogenic substrate are more sensitive than ELISAs with chemiluminogenic substrate. It is the other way around. That is, ELISAs with chemiluminogenic substrates are more sensitive than ELISAs with chromogenic substrates. Then next one. ELISpot measures the number of cells capable of secreting particular biomolecules using a substrate that gives soluble product with enzyme reaction. It is not a soluble product and it should not be a soluble product also. So ELISpot doesn't give a soluble product. So C is also not correct. In Western blot analysis, the product of enzyme substrate reaction localizes at the site precisely where the antibody enzyme conjugate binds to its specific protein band. That's correct. So A and D are correct and the answer is option 2. Then next one. The following statements are made about E. coli SOS repair response to DNA damage. Which of the following option represent the combination of correct statements? That is the question. It is been covered behind the photograph. That's why it is not visible. Okay. Now what is this SOS response? In normal DNA, LEX-A will be bound to the SOS box and SOS will be switched off. In damaged DNA, LEX-A will be cleaved and SOS box will be switched on and DNA damage response occurs. So what actually happens is REC protein, it signals DNA damage. It specifically binds to single-stranded DNAs. On binding with single-stranded DNA fragment, the REC forms a filament-like structure around the DNA. Then a LEX-A repressor comes into contact. LEX-A repressor comes into contact with the REC DNA complex. When a REC DNA interacts with this repressor protein, it converts into REC protease and it is having autocatalytic activity and it will result in the proteolysis of LEX-A protein. Thus, LEX-A protein would not be able to bind to the SOS operator and it activates the inducer protein which repair the DNA damage. So that is the main molecular biology behind SOS response. Okay, we'll see the options now. REC DNA filament complex stimulates the autoproteolytic activity of the LEX-A repressor. That is correct. Then, REC is activated due to the blunt ends of double-stranded breaks caused by DNA damage. It binds to single-stranded DNA, so B is not correct. The SOS response includes the activation of synthesis of translesion polymerases. That is correct because SOS response causes expression of genes that carry out various functions like excision repair, homologous recombination repair, translation, DNA replication and cell division arrest. The destruction of LEX-A promotes synthesis of photolyase which acts along with REC to to reverse the pyrimidine dimer formation process. That is also not correct. So A and C are correct here. So the answer is option 3. Coming to the next question. What is the pH of 10 raised to minus 7 molar solution of HCl? So here in this HCl, the concentration of H plus will be due to concentration of H plus due to acid and concentration of H plus due to dissociation of water. So concentration of H plus total in HCl will be concentration of H plus acid plus concentration of H plus water. We know that concentration of H plus of water is 10 raised to minus 7 mole per liter. Substitute that value and you will get 2 into 10 raised to minus 7. Now calculate the pH. pH is minus log concentration of H plus, and on substituting this 2 into 10 raised to minus 7 into it, you will get 6.79. So the answer here is option 2. Now coming to the next one. ERK5 is a MAP kinase that is activated upon phosphorylation by MEK5. Okay. MEK5 binds with MEKK2 when co-expressed. HEK293 cells were transfected with plasmid encoding ERK5 along with plasmid encoding either MEK5 alone or MEKK2 alone, or both MEKK2 and MEKK5, or both MEKK2 and MEK5AA. 5AA is a MEK5 mutant. Lysates of transfected cells were analyzed by Western blotting using anti-ERK5 antibody as shown below. Okay. So ERK5, it is a MAP kinase and it is activated upon phosphorylation by MEK5. Now if there is MEKK2 also included along with MEK5, then it is going to phosphorylate ERK5. We will see the blot, then I will explain it much more clearly. So HEK293 cells were transfected with plasmid encoding ERK5. Then another plasmid containing MEK5 alone, or plasmid containing MEKK2 alone, or both MEKK2 and MEK5, or both MEKK2 and MEK5AA. MEK5AA, it is a mutant of MEK5. Now these were analyzed by Western blotting using anti-ERK5 antibody. So this is the blot that you got. So you can see here MEK5 alone ERKS. It is not phosphorylated. But if you add MEKK2, then there is some phosphorylation taking place. Only a light band is observed. And whole of ERK5 is not getting phosphorylated. Some ERK5 is remaining without phosphorylation. Then if you see this blot, you can see that MEKK2 plus MEK5 both are required for phosphorylation of ERK5. So ERK5 is phosphorylated and you get a very dark band. No ERK5 is remaining here. But if you use a mutant of MEK5, that is MEK5AA, then there is no phosphorylation. So we will see the question now. From the data in the figure above, the following conclusions were drawn. Now we have to search for combination of all correct statements. Full activation of ERK5 requires both MEKK2 and MEK5. If you see this blot, I already explained that both are required for full activation of ERK5. That is correct. So A is correct. Then next one. Phosphorylation with MEKK2 alone suggests that it can activate ERK5 without MEK5. No, MEK5 is also required for the phosphorylation of ERK5, because if you use MEKK2 you get only a very faint band. So it tells that MEK5 is required. Then difference in the level of phosphorylation with MEKK2 alone and MEKK2 plus MEK5 is due to more phosphorylation at the same site. That is not correct. Phosphorylation with only MEKK2 transfection suggests that it might be associating with endogenous MEK5 to get partially activated, leading to ERK phosphorylation to some extent. That may be a reason why you get a light band over here when MEKK2 alone is used. It's what they have told is that you have endogenous MEK5, that cell whichever you have transfected, it will be having some endogenous MEK5 in that. So this MEK2 whatever you are giving as transfection product will bind with this endogenous MEK5 and then cause phosphorylation of ERK5. That is why you get a faint band over here in your western blot. But actually both MEK2 and MEK5 have to be supplied for complete and full activation of ERK5 phosphorylation. Then next, MEK5AA might be dominant negative mutant of MEK5 which prevents signaling through active endogenous MEK5. What is this dominant negative mutant? These are mutations are those leading to mutant polypeptide that disrupt the activity of the wild type gene when overexpressed. So MEK5AA, it causes disruption of MEK5. Even the endogenous MEK5 is also being disrupted, that you don't get any phosphorylation product in the fourth blot. So E is also correct. So A, D, E are correct and your correct answer is option 4. The next one. The following pattern of gene expression were obtained after activating Notch1 receptor with two different ligands D21 and D24. The following statement depict the correct interpretation of observation. Okay. Now D21, it is one ligand, D24 is another ligand. These are the Notch1 receptors for each of the D1, D21 and D24 respectively. If D21 binds, then HES1 is activated. If D24 binds to Notch1 receptor, then HEY1 is activated. Now we will read the statements. The Notch pathway ligands D21 and D24 both bind to the Notch1 receptor. Yes. But activate different downstream effectors. That is also correct. With similar dynamics. No, it is not similar dynamics. It is with different dynamics. Here you see it is increasing then decreasing, increasing then decreasing. Here it is mostly stable. So it is not similar dynamics. So option wow. So first statement is not correct. Then D21 induces sustained response which preferentially activate the transcriptional target HES1. HES1 transcription activation is correct, but it is not a sustained response. It is increasing, then decreasing, then again increasing. So statement 2 is also not correct. D24 induces pulsatile responses in response to NICD which are required for activating HEA1. It is not a pulsating response, it is almost a continuous response. So 3 is also not correct. The Notch pathway ligands D21 and D24 both bind to Notch1 receptor, okay, but activate the downstream effector NICD with different dynamics. That is correct. One is pulsating, the other is almost continuous. So option 4 is the right answer. Okay, coming to the next question. The following statements were made about the structure of the 30 nanometer chromatin fiber. In the solenoid model, the linker DNA connects the consecutive core particles. So that is a correct statement because the solenoid model is characterized by interaction between consecutive nucleosomes, that is n, n plus 1 and so on. Whereas in the zigzag model, it is interaction between alternate nucleosome, that is n1, then n plus 2 and so on. So, statement A, in the solenoid model, the linker DNA connects the consecutive core particles is correct. In zigzag model, alternating nucleosomes become interacting neighbors. That is also correct. In the solenoid model, 12 nucleosomes are organized into two separate stacks. It is not 12 nucleosomes, it is 6 nucleosomes. Whereas 8 nucleosomes per turn make a single stack in the zigzag model. So statement C is not correct. H1 histone is essentially required as per zigzag model but not as per the solenoid model. Both the model require H1 histone. So that is also not correct. Chromatin fibers prepared with H4 histone that lack their tails could fold into higher order fibers. That is also not correct. So the question was, which of the following options represent the combination of all correct statements? So A and B only. Answer is option 3. The next one. Red-blue color blindness in humans is X-linked recessive disorder. The two parents with normal color vision have two sons. Son 1 is 47XXY chromosome composition and is colorblind. Son 2 is 46XY and is also colorblind. So one son is not normal. The other son is normal, that is why XY, and both of them are colorblind. Assuming that no crossing over took place in prophase 1 of meiosis, Klinefelter syndrome in son 1 resulted due to non-disjunction during which of the following events? Okay, now first we will see whether it is meiosis 1 or meiosis 2. So a non-disjunction occurred during meiosis 1. Then you would not get a normal son because at the end of meiosis 1, that is non-disjunction occurs at meiosis 1. It occurs when the tetrads fail to separate during anaphase 1. So at the end of meiosis 1, there are two haploid daughter cells, one with n plus 1 and the other with n minus 1. But there is no normal son. So it is not at meiosis 1, it should be at meiosis 2. But if it occurred at meiosis 2, you would get a normal son and a Klinefelter's son. So it is meiosis 2. So meiosis 1 option is right off. Then female gamete formation in meiosis 2. It will be female gamete because the X chromosome comes from the mother and the Y chromosome is given by the father. So the non-disjunction has taken place in the X chromosome. So answer is option 2, female gamete formation in meiosis 2. Then next one. A polypeptide was subjected to the following treatment with indicated result. Acid hydrolysis gave this one. Aminopeptidase M didn't give any fragments. Carboxypeptidase A plus carboxypeptidase B also didn't give any fragment. Trypsin followed by Edman degradation of the separated products was this. Which of the following represent the primary structure of the peptide? Now carboxypeptidase A, it cleaves the C-terminal peptide bond of all the amino acid residues except proline, lysine and arginine. Carboxypeptidase B is effective only when arginine and lysine are there at the C-terminal residues. So carboxypeptidases, they cleave at the C-terminal ends. Then aminopeptidase M, it catalyzes the cleavage of all free N-terminal residues. But if you see the question, aminopeptidases and carboxypeptidases, there are no fragments being obtained. This happened because neither N-terminus or C-terminus is free, which means that your protein is circular or cyclic. So cyclic peptide as shown. Answer is option 4. Then next one. Given below are terms related to various techniques in column A and their features in column Y. Match the following. Reverse transcription. Reverse transcription is for cDNA library. Southwestern blotting is for detection of clones encoding nucleic acid binding proteins. Genome walking PCR is for determining the 5' 3' DNA sequence flanking a known gene. RACE is rapid amplification of cDNA ends and it is used in cloning of cDNA ends. So the answer here is option 1, that is A4, B1, C2, D3. Then next one. The following statement refers to factors regulating the fidelity of DNA replication. Which of the following option gives combination of all correct statements? The 5' to 3' exonuclease activity of the replicative DNA polymerase. That is not correct because replicative DNA polymerase is DNA polymerase 3 and it is not having 5' to 3' exonuclease activity. Then next one. Imbalanced intracellular concentration of the 4 DNTPs. That is correct. Increased intracellular concentration of RNTPs resulting in increased incorporation of RNTP during DNA synthesis, which are not easily removed by the polymerase's proofreading activity. That is also correct. Removal of incorrectly incorporated nucleotides by the mismatch repair system. That is also correct. So B, C, D are correct, whereas A is the only one that is wrong. So answer is option 2. Then next one. Which of the following statement is correct in the context of erythromycin mediated inhibition of protein synthesis in bacteria? So erythromycin, it binds to the 23S rRNA molecule in the 50S ribosomal subunit. It results in peptide chain elongation prevention. The binding blocks the exit of the growing peptide, thus inhibiting the translocation of the peptides. Now we will see what they have mentioned. Erythromycin inhibits protein synthesis at the step of elongation, but is nonetheless allowed translation of the first few codon. That is correct. Erythromycin inhibits protein synthesis at step of elongation, okay, and it prevents formation of even the first peptide bond. That is not correct. Erythromycin inhibits formation of the translation initiation complex. It is not initiation, it takes place at the erythromycin is binding at the elongation time, not at initiation time. So erythromycin is toxic to bacteria because it results in initiation. Again initiation, it is not initiation, it is elongation. So only one is the correct answer. So answer is option 1.